everyone. Good morning. It's for me a pleasure to be here. Thank you to the Computational Biology and Bioinformatics Hub and all of you for being here. I'm Eduardo Andrés León. I'm from the Bioinformatics Unit at the Institute of Parasitology and Biomedicine López Neira in Granada from CSIC. You have here my X account, Blue Sky, and also my email if you want to talk to me or whatever. I'm going to talk about a tool that I developed years ago during my PhD in the part of Seville, in this case. It's called miRNA-Seq and RNA-Seq multiprocess analysis. You see here some of the acronyms. And as I was in Seville in that moment, I used this name, miARma-Seq, as the real name. miARma-Seq is a comprehensive tool for microRNA, messenger RNA, and circRNA analysis. This was published in 2016 during my stay at the IRIS in Seville in the Ana Rojas lab. This was published on scientific report. This publication, according to the mention, is extremely high cited and it received around 13 times more citation than other similar publications on the same age and on the same area on average, which is a lot for us. It was a success. It has a lot of citation, as I said. So we were invited in 2019 to do an improvement of this pipeline. In this case, we perform a simultaneous integration of data from microRNA and messenger RNA, and it was published in the Method journal. Also, this publication is highly cited and received around 4 times more citation than the average. So when we developed miARma, our aim was to develop a tool for those researchers who knows nothing about bioinformatics, about command line interfaces, biotheme, also transcriptome analysis or Linux computers. Also, it was important for us to provide a tool where all the software, or at least most of the software, were already provided and installed and compiled. And the whole idea was also to perform automatically any transcriptome analysis from the very beginning, doing a quality analysis of the sequencing file to the functional enrichment to see biological process or pathway altered during the analysis due to whatever you are studying, a disease, knockout, or whatever. This is a workflow of how miARma works. We have the part of microRNA where we can able to study known microRNAs. We use data from the MIR-based database, but also you can perform the identification of new microRNAs in any organisms, even on those organisms where you know anything about microRNAs. You can also study other kind of RNAs like messenger RNA, long-coding RNA, and even circRNA. The software is divided in modules, and also the way to run the software is also can be configured in modules. So the first module is the quality module. It uses a software called FastQC that creates reports in web pages where you can study the quality of the sequence, contamination, adapters, whatever. Okay, so the second module is mainly specific for microRNA, but also can be used in RNA. As you know, microRNAs, the major ones, are around 23 size long. So if you are sequencing, I don't know, 50 nucleotides, 75 nucleotides, 100, 150, which are the regular numbers, most of the sequence are no microRNAs, are adapted. So you have to remove the adapter in order to get these 23, 24 nucleotides, which are the real microRNAs. So you need to remove the adapter. The idea is to know what adapter has been used by your genomic unit or the company where you have done the sequencing. They should provide to you, but also see if you are using data from an article. Maybe they don't include the adapter, so we use a software to predict this adapter. When we have the adapter sequencing, it can be removed by these two software, QuickAdapt or Reaper. Then the next module is the aligner module. For the novo prediction and quantification of microRNAs, we use miRNAs. For microRNAs, we mainly use both type 1 and both type 2. For circular RNAs, we use DWA. And for messenger RNAs, you can use both type 1, both type 2, ISAT, STAR, even the combination of some of them. So once you have all your sequence aligned to your reference genome, the idea is to count these reads into exon, into genes, into transcriptome, into whatever you want to analyze. We use feature counts in most of the analyses, but in the case of circular RNAs, we use Siri, and in the case of the novo microRNA, the software miRNA does create this count file. Once we have this count file, that is the number of reads for each feature, we can perform a differential expression analysis. You can select among HR or non-seq. The idea is to compare different types of samples, your reference or your control, your healthy sample against your disease, your mutation, whatever you are studying, in order to see the difference at the transcriptome level between the both groups. Also, you have another module, which is the functional module. So if you are studying messenger RNA, at the end you will have here differential expressing genes. So the idea is to look for pathways in the case of KERT or gene ontology terms, molecular function terms, biological process terms, related to these changes among your groups of study. Okay, so you have also these results and some graphs in order to see these differential expressive genes associated to more general biological process. And then we include the target prediction module, which was included in the second article, in the one published in the Method journal, in order to look for prediction or relation among microRNA inhibiting the expression of messenger RNA using a database that I also developed during my PhD, which is called miRNA. So first, in order to find miARMA and to install miARMA, as I said, the installation is quite straightforward. So you have here the link in order to download the article. You have also the QR. So the idea is to just download a zip file and compress the zip file, and that's all. You have installed ready to use. For Unix machines such as Linux, FreeBSD, LGD, or MacOS, the only thing you need is bioconductors. So this is like the prerequisite that miARMA miRNAs. And if you are using Windows, we provide a Docker image with everything installed. So as I said, it's quite easy to have miARMA up and running. Once you have miARMA downloaded and configured, this is more or less the output that you will see. When you start the analysis, what kind of analysis are you doing, which module are you going to process, and for example, in the case of the first module, which is the quality, when they start, when they finish, and more or less this is everything. Okay. So the input, how miARMA works, how miARMA needs your FASTQ files, and how it processes. We did this as easy as possible. So we use a configuration file, which is a ini file, in this regular standard for configuration for any kind of software. So we have here a section which is called general. This is a mandatory section in order to select which type of analysis are you going to process. In this case, it's a messenger RNA. To verbose in order to get the idea what miARMA is doing. So, for example, this is with a verbose 0, so you only have information about what it's doing, but you can have a lot of more information. So, which file I'm processing at the moment, and this kind of thing. So you can modify the verbose to a different value, different whatever, that is not 0. Then you have to provide the path where your sequencing files are in FASTQ file. How many processes do you want to run at the same time? This will depend on your computer. If you are using an HPC, you can use, I don't know, 64, 128. A label for the analysis. In this case, we use ASMA. So we will have most of our result in the name of the files as the output, this word ASMA, and also in part of the graph that we develop. This is also the path where miARMA has been installed, or in this case, uncompressed. Okay? This dot means that this ini file is in the same folder where miARMA has been uncompressed. Output directory, so where I want to store all the results, which organism I'm working with, and what type of sequencing. As you may know, the only two options, single end or paired end, where you are sequencing two parts of the messenger RNA. And also the strand. Maybe you are using a assay which is strand specific. In this case, you use yes. If you are on the reverse strand of the DNA RNA, in this case, you can provide reverse. Or if you are using if you are not doing a strand specific assay, you just provide no, as this in the example. So next, once we have this general section, the mandatory one, we start with the first module, which is the quality module. In order to ask miARMA to do this quality analysis, is just to include these two lines. A new section, which is called quality, and this prefix equal to pre, in this case. You have three possible values, pre, post, or both. Pre means that you read the FASTQ file, you analyze this quality, and that's all. We create these HTML files. And if you are removing the adapter, and the idea is to create this quality analysis at the beginning, then remove the adapter, and then perform again another FASTQ file, another quality analysis, in order to see if these adapters have been removed. So in this case, you can use post only to do this second quality analysis, or both, which means to do the pre at the first moment, to remove the adapter, and then to create the second analysis. That's an example. For example, we are seeing here, which is the quality of the reads, which is quite good. And then we have another section where we can ask to see how the adapter is in our sequence. We see here a huge content of adapter from the position 12 to almost the end of our sequence. So this is quite bad for our data. So the idea is to remove. So in this case, we suppose that we know the adapter. Okay, so we provide adapter. We provide the software to remove this adapter sequence in this new section called adapter, and we obtain also a graph once the sequence has been removed. In this case, as you see, it's a microRNA analysis. So we are expecting most of the size of the read to be related to 22, 23, or 24, as is the size of the major microRNAs. And what happens when you don't know the sequence? So this is how to perform an adapter removal when you don't know the adapter sequence. Again, you include the adapter section, you provide the software, and as you see, we are not including any adapter sequence. We are just provide that we don't want reads shorter than 18 size, 18 length, kind nucleotides. That means when the sequence is small, it can be aligned to different parts of the genome, and this is something that you don't want. And also, if we are studying microRNA, which is, as I said, around 22, 23, so we are giving a window of 18 to 26. So this all reads greater than 26 or smaller than 18 will be removed. But we also, if you want to provide a quality, a meaning quality in order to analyze this read, you can provide with this parameter, mean quality. In this case, it's 25, and you know it's from 1 to 40, so 25 is a really good number. So if the quality of the read is not good enough, we are not going to use it, and this will be removed. And then, as we are going to use a software to predict the sequence, how many predictions do you want to be created? And this is the standard value of the software. The software is called miNION. miNION reads the FASTQ file. It reads the sequence at the edges of each read, and it looks for consensus sequence. So that's an example. For example, in the 3', we have always more or less the same sequence. Also in the 3', so we can assume that this yellow sequence is the adapter sequence that we want to remove. But what happens when we have a really high overexpression of a sequence? Something that we have seen in some of our experiments. So when the overexpression is huge, there is a high probability to obtain an adapter sequence which is related with the sequence of this biological feature. So to avoid this, we use miARMA directly connect to blood, to the UCSC. It takes the sequence, it looks in the genome if this adapter sequence is inside the genome or not. So if the sequence is within the genome of our organism, we assume that this is not an adapter, and if we don't have any hit, we assume that this is a good adapter. So when we have checked this, we obtain the real a good adapter, so we can remove it. So this was as well our result of the pre-quality analysis. If we compare with the post-quality analysis, we must see something like this with this green check. So this means that we have no adapter, and we can proceed with the next step. The next step is the aligner module. It depends also on the type of analysis. So, for example, if you want to make a de novo identification or quantification of microRNA in an organism. We use the miRDIP2 software. We perform automatically the adaptive removal, the alignment, the microRNA identification, and even microRNA quantification. This tool was developed by the Rachesky lab. It was really high published, and it is used a lot. So how do I create this? How do I say miARma to perform this de novo quantification? So you can create again a new section. miRDIP2 only use both I1, so it needs the genome to be indexed. So when we try to align this sequence against our genome, we use the index to be as faster as possible. If you know that after you can include it, if not, nothing happens. miRDIP can also work without the adaptive removal. If you know some of the microRNA or your organism, you can provide here with this parameter, and also the gene, which are the precursor of these microRNAs. So in that sense, miRDIP will look for this noun microRNA. This will be also quantified, but also will look for another non-noun microRNA, and this will be also quantified and included in the final report. And also you need the sequence of your genome in FASTA format. Okay. In the case of the other analysis, it's just an aligner, so more or less the configuration is quite similar. So you include the section which is called aligner. You select which aligner do you want, both I1, both I2, PWA, miRDIP, top hat, CSAT, start, also combination, for example, both I1 and both I2, okay, and this will be aligned with these two aligners. And also you need to include the index for the aligner you have select. So this is an example for a start. This is an example of CSAT. I change here the aligner, so I change here the index of the genome. An example we put I1, and also we include some of the most interesting feature or more used feature in each of the aligner. For example, in this case, we are saying that we don't want any mismatches in the seed alignment among our read and the reference genome, and the length of the substring in the seed, which is a value among 5 and 32. We want the number 19 in this case. But you can also provide other parameters to include in the analysis. So you have here the both I1 parameter, and you can include each of the parameters you want. In the case of both I2, this is both I2. We have also stat parameters, CSAT2 parameters. So it is more or less always quite similar. Another feature that we include, which was really useful for us, is maybe you don't have the index, but it's quite easy to download the genome of your organism. So you can provide the name of the aligner, where the path to the FASTA file of your genome, and an index name. So in this case, CSAT will take this FASTA the genome and will create automatically the index. So you can include here both I1, both I2, start, BWB, A, just the FASTA, just the name. miARma will create the index, and then it will continue with alignment of the sequences, just in an automatic way. So when we have our aligner, we have to quantify the expression of the genes or the exon, whatever you want. So in this case, we have a special section, which is read count. So in order to know where do you have to count, you have to provide the annotation of your genome. You can provide a GTF file and also a GFF file. This file is a tabular file with different columns. So in our case, we always count reads which are aligned to the exon, and then we summarize this into gene names or even in transcript names. So if a gene has 4 transcripts and you use transcript ID, you will have 4 different results with the different reads or different count of the reads aligning to the exon for the transcript 1, for the transcript 2, 3, and 4. And if you provide here gene ID, you will have just one feature, and this will be the sum of all of the transcript ID. So we will count all reads aligned to the exon, and we will quantify and summarize into a gene feature. Okay. This name, which is gene ID, transcript ID, depends on the format of your annotation file. It could be gene ID or gene name. You just have to look for a couple of lines, and you will see which are the proper feature to include here. Also, we include this parameter, which is the quality. The idea is to avoid counting low quality reads. And you can also provide other parameters to the feature count software. In this case, these are the ones that we always include, which is count reads in the parent sequence. Only reads with both of the pairs are aligned. Avoid multi-mapping. These are the reads which are aligned in different parts of the genome. Include overlapping gene reads. That means if a read is shared among 2 different exons, for example, this will be counted in the 2 exons. And this is to avoid chimeras. A chimera is when one of the pair of the read is aligned to one chromosome, and the other pair is in another chromosome, which is quite strange. So we don't want to include these strange things. For example, in order to quantify circRNAs, it's more or less the same. We have to provide the database, which is your annotation file, again, GTF or GFF, the genome of your organism with the FASTA parameter, and then which method do you want to use to quantify and identify the circRNA. At the moment, miARma includes 2 of them, which is CD1 and CD2, which were the software that was published more or less at the same time of miARma. It includes several experimental validation, and it works really well. So that's the reason to include this software. Okay, so once we have quantified or identified all our features, the idea is to compare. In a transcriptome analysis, you always compare samples. You have samples from a regular cell line, a healthy person, or whatever. And then you have the other group, which is the patient having a treatment or a cell line with a mutation or whatever. And the idea is to look for differences in the transcriptome of the 2 groups. So in order to do this, you can select 2 different software, which is HR or miARma-Seq. So you include the differential expression analysis section. We have a lot of parameters. I will explain more or less each of them. Then the software, we have here two files that we'll explain later. Filter, that means that we don't want low expressed genes to be included in our analysis. This is a cutoff to say to miARma how do we take into account this low expressed gene. So in this case, a low expressed gene will be the one which has a CPM value of 2. What does mean this? For example, if you have 10 million results, 10 million reads, so This means that you will need at least 20 reads assigned to your gene in order to be a non-low expressed gene. If you have more than 20, it's a regular expression. If you have less than 20, it will be removed. How many of our samples do we need to be low expressed in order to remove a gene? We always suggest the minimum value of our group. If you are comparing 4 healthy people with 10 disease people having whatever disease, the smallest group is 4, so you have to include here 4 and more or less like this. Which method to normalize? And this software challenge has several methods included. We always suggest TMM if we are including replicate, and then we have a file with a normalized matrix of expression for each of the samples. So we create a CPN normalized expression file, and also a RPKM expression file. This file, the target file and the contrast file, as you say, we have never said how many files we have, how many samples, what kind of sample. We have say nothing about this in the whole analysis. So this is the place to give this information. So you have this metadata file, which is the target file, and you have the name of the file, which is the file name with your FASTQ file, the name that we will use for the different graphs that we are going to include, and the tag. So here we have 3 tags which are resistant to a drug, and 3 of them which are sensitive to this drug. This is the way to give some information about your sample. So here we are analyzing just 6 samples. And the control file is where you specify which type of comparison you want to perform. In this case, you want to see the resistant against the sensitive. So high expressed gene will be those which are high expressed here, and low expressed gene or inhibited gene will be the ones that are low expressed here, which also means that this will be the one that are high expressed here. Okay. So that's the way to perform all the analysis. In the case of miARMA-Seq, it's quite similar. We provide the name of the sample, the name of the software, the files in order to see which file do you want to analyze, which kind of analysis you want to include. In this case, it's like cutoff, so we want the really good differential expression gene, which is at the 90%. Also, I want to filter with filter method, with CPN value. So this is more or less the similar. Here, the TMM algorithm to normalize, the replicate, with type of replicate are we including, and also the CPN or the RPKM files to perform some graphs or some manual analysis. This module also creates several graphs. In this case, is the normalized log2 counts, which are the ones that are aligned. The idea is to see that more or less this line, which are the median value, are more or less similar. I mean that you can compare among the samples. We perform also some clustering. This is a dendrogram of our sample. The idea is to have one of your group here and the other here, in order to see that are different enough to be compared. Multidimensional scaling plots, PCA plots, heat map plot, and this kind of thing. And finally, the final result, which is an Excel file with the gene name, the length of the gene, the fold chain, which is to see if the gene is overexpressed or uninhibited. This minus implies that this gene has been inhibited, and this, which is a positive, means that this gene is highly expressed. And as the fold discovery rate, which is the statistical value, is less than 0.05, that means that this fold chain is quite reliable. So these two are the ones below this regular score, we can say that these are our two differential expressed genes. One is overexpressed and one is inhibited. So with this differential expressing gene, we can look for functional analysis in order to see if they are related to some of the biological process. In this case here, we have the gene ontology category. We have the term, which is RNA processing, RNA splicing, whatever. These are the ontology here to be filtered, biological process, cellular complex, molecular function, and the p-value to be overexpressed, which is this is high enriched term, or underrepresented term, with this related to the number of your differential expressed genes. Some validation. The idea is to see if miARma is working good or not. So we download data, in this case from Camps et al., and we analyze this data using miARma in a different way that Camps et al. Did. And we more or less have the same fold chain, so we are doing quite well. This is for microRNAs. In the case of the messenger RNAs, we use the Camps et al. Article. We download all the FASTQ file. We analyze with miARma. As you see here, again, we are doing very well. Camps et al. Did some validation using RT-PCR, qPCR, and again, our log fold chain are quite good, so we can say that miARma is performing really well. And in the case of circRNAs, we use the data for Pan et al. They provide the counts for the identified circRNAs and also validated in experimental validation. So we are more or less doing quite similar to them. So it's a way to say that miARma is doing automatically without including of parameters. It's just with the configuration file, and miARma does everything, and we are doing very similar to other authors. We did some improvement for the second article. We include this target prediction module, also some statistical correlation. As you know, microRNA downregulate microRNAs, so we look for anti-correlation, which are statistically significant using either Pearson or Spearman. So this is data that we use for an active medicine article, which is a low-coding RNA, which is this one, MIR100. And the idea in this article is to study colorectal cancer cells. Some of them are sensitive to a drug, which is called cetuximab, and some of them are resistant. So they perform a transcriptome analysis of RNA-seq, and also for microRNA features. So this is the whole INI file that we provide. This is our idea to reproduce the data. You can use this miNAP file, which is included in our GitHub also, and you will perform analysis the same way as we did. We have the general section where we provide what are the risks, how many threads, what can do we want to store the results. These are Pirate N, which are not stranded. We will do quality analysis. We align with GitHub, and we use the read count to quantify just high quality reads, avoiding chimeras and multi-mapping, including overlapping and risks which are both aligned. We use the chart to the differential expression analysis. All of this is for the messenger RNA. For microRNA, more or less the same. You see here the difference with the adapter. We asked the author for the adapter sequencing. They never replied. So we try to use Minion to get to predict this sequence. It predicts quite well. We align using both Taiwan. We quantify. We do the differential expression analysis in the same way, and then the idea is to look for differential expressing microRNA, which are regulating differential expressing genes. So this is the folder of the differential expressing gene, which is in the output folder of the previous analysis. And this is more or less everything. miARma creates volcano plots of the most expressed or inhibited genes. In this case, you can see this gene which is highly overexpressed. Okay, you have also another high expressed gene, and in this case, the low expressed genes or the inhibited genes. Also for microRNAs, we see 4 microRNAs which are quite overexpressed. This is miR100 and miR125, even in the 2 sides of the sheet, and also the LED7A. So we have 3 microRNA highly expressed, taking into account that this comes from the same gene. So we can say 3. Why this happened? This happened because I'll tell you later. We also look for the regulation or the correlation among the expression of these long non-coding RNA and the expression of each of the major microRNA. So if this long coding increase, also increase the expression of this microRNA. This is the same for 125 3P, 125 5P, and also for the LED7. So this happened because this is the gene, the long coding gene. It has 4 exons. So among the exon 1 and the exon 2, we have the genes of our 3 microRNA. So if we are expressing a lot this long non-coding, we are expressing a lot also these microRNA. So as you see, if we create a heat map, we see that in the resistant sample, we have a high overexpression of this long non-coding and also to these 4 microRNAs. We will look for regulation among these microRNAs and these high expressed genes, and we see some really interesting regulation among high expressed gene and low expressed microRNA, and the other way around. And most of these genes are related to beta-catenin, which are related to the resistance to this rituximab. So in order to finish, I would like to say that you have more information and the whole documentation in the miARma GitHub webpage. You have these 3 PDF files. Also, you have some example. In this case, for non-microRNA, you have here the 6 modules. Inside of each folder, you have different ways to use of the parameters, and we provide also a pipeline for a whole microRNA analysis. The same for messenger RNA, all the modules with different examples in each of the folders, and also we provide a whole pipeline to analyze with HISAT, STAR, or even TORHAT, very similar to circular RNAs. And to finish, I would like to say that the latest version of miARma was 1.75, but we have also a new version of miARma, which is the miARma version 2, but we have renamed this software, and now it's called reanalyze GCA, which it will be the next talk by Jose Luis. So thank you very much to all of you, and thank you for your attention, and it will be a pleasure for me to answer all your questions. Thank you very much.